The two basic phases of protein synthesis, transcription and translation, are the same in prokaryotes and eukaryotes. How prokaryotes and eukaryotes go about those two processes, however, are different. To understand how protein synthesis differs in eukaryotes compared to prokaryotes, let's dive into video 12.5 on eukaryotic gene regulation. This video will focus on these goals. Pause for more details. The operons video explained how prokaryotes are built for speed. Prokaryote adaptations for speedy transcription and translation include using polyribosomes to transcribe and translate a gene many times and all at the same time. Likewise, operons have clusters of genes that code for proteins with related jobs, so all the proteins needed for a job are all transcribed and translated all at once. Also with operons, repressor proteins are always ready to quickly bind or unbind operators to turn a gene on or off. Eukaryotes, though, have different needs that require different gene regulation. Many eukaryotes are multicellular. Remember, each cell has the same exact full copy of the DNA, and in humans, this means that each cell has all 46 chromosomes, but each cell certainly isn't identical. Making different cell types from starting stem cells is called differentiation, and different cell types do different jobs, and doing those different jobs requires different proteins that are made by turning different genes on and off. Let's slow down on that for a sec. All right, so here are three different cell types: blood cells, muscle cells, and pancreatic cells. And the lines underneath each of those represent different genes that are turned on or off in each cell so that the cell can do its job. Now, genes can fall into two basic categories: housekeeping genes and luxury genes. And just like housekeeping is something that we all have to do, like laundry, taking out the trash, sweeping up the floor, there are certain jobs that all cells have to do. Remember how all cells make ATP and all cells make proteins? Well, the genes involved in those basic housekeeping jobs are turned on in all cells, just like you can see right up here. Housekeeping genes are turned on in all cells. Now, luxury genes are a different story. Differentiated cells have differentiated jobs. Red blood cells carry oxygen, muscle cells contract, and pancreatic cells make digestive enzymes and hormones. And those specialized jobs require specialized luxury proteins. Red blood cells need high levels of hemoglobin, muscle cells need a lot of the contractile protein myosin, and pancreatic beta cells have high insulin expression. So in other words, certain cells have high expression of certain luxury genes to do their job, but luxury genes that aren't needed get permanently turned off in cells that don't need them. Now, besides differentiation, eukaryotes regulate gene expression in response to signals like you can see out here. So in this plant cell, it's responding to light from the environment and calcium released by other cells. Now, light activates phytochrome molecules and calcium enters through calcium channels, and both signaling pathways activate relay molecules that end up activating transcription factors. And transcription factors bind a certain gene in the DNA and attract RNA polymerase to that gene to increase transcription. Now, in this case, the end result is production of proteins that help plants do the greening response, where they make more chloroplasts. So eukaryotes regulate their genes for cell differentiation and to respond to their environment. But how exactly does gene regulation happen? Well, because eukaryotic protein synthesis is uncoupled, with transcription happening in the nucleus and translation happening out in the cytoplasm, there are lots of opportunities to hit the pause button or stop button on protein synthesis in eukaryotes. I'm going to focus on three main types of eukaryotic gene regulation. The first is structural control, where DNA itself is more or less accessible to RNA polymerase in certain genes. And the second type of regulation is transcriptional control, where the levels of mRNA being made can be adjusted. And finally, post-transcriptional control regulates how long an mRNA or proteins stick around. Let's talk about each one of those more. To fit in the nucleus, DNA is organized and packed up on histone proteins, but For DNA to be used, we know that DNA needs to be diffuse or unpacked. And here's the cool thing in eukaryotes. DNA can be unpacked in spots where genes are needed, like housekeeping genes, but packed up for luxury genes that will never be needed. Now, there's terms to describe this. Condensed regions of the DNA are called heterochromatin, and heterochromatin is tightly packed up so the genes can't be accessed by RNA polymerase for transcription. In contrast, DNA that's spread out and accessible is called euchromatin. And I'll relate this back to our mnemonic from before. We said diffuse DNA is DNA that is being used. And another name for this is euchromatin. Hopefully that's helpful. Now, DNA is packed or unpacked by adding two different chemical groups, acetyl groups and methyl groups. When acetyl groups are added to histones, like here, here, and here, the DNA changes from heterochromatin to euchromatin. Then the DNA, once it's all spread out, can be transcribed and protein can be made. Now, if the gene isn't needed for a bit, the histones can be deacetylated, where these acetyl groups are taken off and then pack up that gene for later use. Another way to pack up DNA is by adding methyl groups, CH3, to the DNA itself in a process called methylation. But there's a twist. With methylation, DNA packing is permanent. So methylation is good for luxury genes that will never be used, but genes that might be used later on, definitely don't want to methylate those. Now, even if a gene is unpacked, doesn't mean that gene is turned on all the time. First, RNA polymerase has to find the gene and make mRNA copies. And transcription factors and activators are proteins that attract RNA polymerase to a gene so the gene gets transcribed more. Activator proteins bind to enhancer DNA sequences, and transcription factors bind to the promoter. Now, both of those increase RNA polymerase binding and then gene transcription. Similarly, repressor proteins can block RNA polymerase from transcribing a gene. Here's a look at those activators and transcription factors at work. Activator proteins in red are bound to enhancer DNA sequences in yellow here, here, and here. And similarly, transcription factors shown in green are bound to the promoter DNA shown in dark blue. Now, together, you can see that these activators and transcription factors are stabilizing and touching each other and stabilizing and touching RNA polymerase, then attracting RNA polymerase to the promoter to keep making mRNA. Now, even once that mRNA is made, there are still opportunities for gene regulation in eukaryotes, and one of those opportunities has to do with the life cycle of an mRNA. Now, if an mRNA is around, I want you to imagine that mRNA is just going to be translated and translated and translated again by ribosomes, but the lifespan of mRNA is influenced by a few different factors, and one of those factors is the poly-A tail. The poly-A tail is a post-transcriptional modification of the pre-mRNA that's added before the mRNA leaves the nucleus. Here's another view of that poly-A tail. Now, a poly-A tail can be longer or shorter, and this diagram also shows how the poly-A tail influences the life of the mRNA. In the cytoplasm, there are RNase enzymes that degrade mRNA by eating away the poly-A tail. And once the RNase gets to the coding segment of the RNA, shown here or here, the RNA isn't useful to make the protein. It doesn't have the whole code anymore. So the longer the poly-A tail, the longer that mRNA sticks around in the cytoplasm, and the more protein can be made from that RNA code. Besides the poly-A tail, an mRNA can be shut down using RNAi, or RNA interference. Here's how RNAi works. Specialized types of RNA are made called small interfering RNA, or siRNA. siRNA are complementary to specific mRNAs, so they'll stick together, the siRNAs and the mRNAs. In order to work, though, an siRNA has to be joined with proteins to form a RISC complex, or RNA-induced silencing complex. Now, the RISC goes around and tests RNAs that are out in the cytoplasm, and if the RISC finds an mRNA with a complementary code, that mRNA will be chopped up and no more proteins can be made from that mRNA, turning the gene off. Scientists are finding that RNA interference, is actually more nuanced than previously thought. It was previously thought that an RNA would be completely chopped into bits. So if the siRNA and the RISC perfectly matches the RNA, then the RNA is chopped up like on the left. But siRNAs can be made that only partially match the mRNA, and in that case, the mRNA is only stopped temporarily. It's reversible. Pretty cool. The last opportunity to regulate proteins that I want to talk about comes even after the proteins are made. When a protein has served its function, the cell destroys the protein using a proteasome, shown in yellow right here. The proteasome is a big old complex that forms this like trash barrel shape. And for a protein to get added to this trash barrel proteasome, the protein must be tagged with another protein called ubiquitin, as shown here. So here we go. This purple protein isn't needed anymore. So the protein is ubiquitinated. Ubiquitination is the sign that the protein should go into the proteasome. The proteasome hydrolyzes this protein into peptides, which are then hydrolyzed into amino acids, and then the amino acids and the ubiquitin can be reused. Woo, that was a lot, and you thought operons were complicated. All right, here's a recap. Eukaryotes need gene regulation to form differentiated cells, and then for those cells to respond to signals they receive. Eukaryotic gene regulation takes advantage of the breaks in the stages of protein synthesis in eukaryotes. DNA packing is the earliest opportunity to increase gene expression by unpacking DNA or decrease gene expression by packing DNA. Next, transcription factors and activators can attract RNA polymerase to a gene to increase gene expression. And then the lifespan of mRNA is another possibility for gene regulation, where a longer poly A tail can result in higher gene expression, and RNAi can shut things down in a hurry, temporarily or permanently. Finally, when a protein is no longer needed, the protein can be tagged with ubiquitin and degraded by a proteasome.